Thank you for standing by and welcome to today's Chimeric Therapeutics investor webinar, discussing today's announcement of positive preliminary phase 1 data for chlorotoxin CAR T in recurrent brain cancer. All participants are in a listen-only mode. There will be an initial presentation lasting for approximately 15 to 20 minutes, followed by some Q&A. If you do have a question you'd like to submit, please feel free to type it in using the Q&A function within Zoom. Presenting on behalf of Chimeric today, we have the CEO and Managing Director, Jennifer Chow, and Chief Medical Officer, Dr. Jason Litton. To begin, I'll hand it over to Jen. Please go ahead.
Thanks so much, Matt. And thank you, everybody, for taking the time this morning to join us on very short notice to discuss the positive data that we are now seeing with CHM 1101, our chlorotoxin CAR T. I can't tell you personally how excited I am to be able to have this investor webinar today to bring everybody up to speed on this data and really to make good on the commitment that we made with this asset to bring this data to you. So without further ado, I am going to just, take you through a few introduction slides, and then I'm actually going to turn it over to Jason, our Chief Medical Officer, to take you through the data. And at the end, of course, happy to take any questions that you have. So this is the summary of the data that we actually announced this morning. And you'll see it here characterized in 3 different categories. And Jason will spend some time taking you through each of those. The disease control rate, meaning how many patients actually achieve disease stability, the survival that those patients achieved, and then the safety that we saw with it. Across all 3 measures, we were extremely pleased with the data. The disease control rate of 55% exceeds the historical controls for assets that the NCCN guidelines actually recommend. And those are about 20 to 37%. So very pleased with the 55% disease control we've seen. 10-month survival in patients that achieved disease control was something that very much was beyond what we actually expected, and we are very pleased. And really, Jason will take you into more detail about this, but we actually have 2 patients that actually have gone beyond 14-month survival, one who is ongoing and alive. So this is also a very important milestone for us, particularly knowing that patients generally only expect to survive about 7 months after their first recurrence of this disease. And then the primary endpoint of this trial being safety, we were very happy to see that this therapy was generally well tolerated and there were no significant safety concerns at all. So that's the data in a summary. Just to set the landscape for everybody and for Jason's, reveal of the data in a little bit more depth, I want to share a little bit about glioblastoma for those that aren't as familiar with the disease area that CHM 1101 is actually treating. So glioblastoma is a brain cancer, and it really is the most deadly of all primary brain cancers. It grows very fast, and unfortunately, survival for patients that are diagnosed with glioblastoma is very poor. It is a fairly rare disease. In Australia, there's only about 1,000 new cases of it diagnosed every year. And generally, it's more common in men. And average, you see patients present with glioblastoma in their early 60s. There aren't very solid risk factors that we can point to and say, hey, that's the reason why someone has glioblastoma. The majority of risk factors are actually unknown, unfortunately. So what becomes the prognosis? Well, if a patient is diagnosed with glioblastoma and actually chooses not to be treated, they can expect to survive about 3 months. With treatment right now, we expect to see survival somewhere in the neighborhood of 15 months from diagnosis. And in Australia, what we actually know is that the 5-year survival rate for these patients is only 4.6%. So unfortunately, this disease is one of the most deadly diseases out there, and these patients are very challenging to treat. When we go to treat these patients, though, although the slide is a little bit messy, I want to take you through it because it's important. Treatment becomes more challenging as the disease progresses. So if we start on the far left of this slide and a patient is diagnosed, what you see down below is generally the survival expectation of about 15 months. When a patient is diagnosed with glioblastoma, they're generally offered a standard of care therapy. And that's a therapy that is recognized worldwide as providing the best possible outcome for these patients. The standard of care therapy in first line, and that's really when you have your first therapy for glioblastoma, is surgery, radiation, and a drug called temozolomide or Temidol. And generally, what we know is that after maybe 7, 8, 9 months, patients will actually recur or their disease will progress. And they'll move on to the right-hand side of this screen where we start to offer them second line or third line, fourth line therapies. And what we do know there is that the minute the person or a patient progresses and enters second line, their overall survival expectation is only about 7 months. And as they move from second to third and third to fourth line, they become much more difficult to treat. So certainly a very challenging patient population to treat, and honestly, no great therapies in recurrent glioblastoma to date. So with that as a little bit of a background, I'm going to invite our Chief Medical Officer, Jason Litton, to talk to you and give you a little bit more depth on the data that we actually announced today. Jason?
Thanks, Jen. That provides a really great framework for us to continue this discussion and why I'm actually really excited to take you through our data. So on the next slide, I'll start to provide some of the details on the patients that were enrolled in this clinical trial. As shown here, 46% of the patients had received CHM 1101 as fourth line therapy. And there was an additional 18% that received it as fifth line therapy. So almost 2/3 of the patients were heavily pretreated and would be expected to have very poor outcomes from any treatment they could be offered. On the right side of the screen, we have some of their demographics listed, where the mean age of patients treated in this study was 57 years, and the range was 37 to 73, with a slight male predominance, as Jen mentioned, is usually the rule. These patients, most of whom had had prior surgeries, and some had had several prior surgeries for their disease. And all of the patients had grade 4 or the highest grade of brain tumor, one with an astrocytoma. On the next slide, we can start to talk about, and Jen started to kind of lay out for us the way we think about response to treatment and what a new drug for patients with glioblastoma really has to demonstrate in order for it to be treatment changing and disease modifying. So the first thing that we look at is a response, an assessment of whether or not a patient responds to a therapy. And we evaluate that in two different ways. First, the number of patients who have had a response, how many, and second, the depth and duration of that response, or how strong is that response in those patients who have responded. We also care a lot about safety. Obviously, these are patients who we don't want to do harm to, and we want to provide medicines that are safe and have the potential to generate benefit for them. And so we look very closely at safety, especially in a first-in-human phase 1 study, and specifically look at what we call adverse events or negative things that a patient might experience while they're receiving the drug, whether or not they actually have to do with that drug or not. And then, most importantly, we look at the severity of those events. And we look both at the very severe events, and there don't need to be very many of them for us to pay very close attention. But then we also look very closely at the lower grade events that can be really problematic for patients over time. And that's a really important assessment that we take forward. But really, in glioblastoma, where survival and outcomes are so abysmal, it's really survival that is the gold standard for what we're looking for. And survival is plainly exactly what it sounds like, an assessment of whether or not the therapy helps a patient live a longer life. We look at that in a couple different ways too. How many patients have improved survival with the therapy? And then specifically, and what we'll talk about in a few minutes, is how much longer do these patients actually survive after they receive this therapy? So on the next slide, I start to try to put into context what we demonstrated in this study as it compares to approved drugs that patients with glioblastoma are receiving today. So in the middle column, you'll see CHM 1101 was largely treating patients in late line, fourth and even fifth lines of treatment, and demonstrated disease control in more than half of those patients, 55%. Lomustine capsules and Temizar or temozolomide are both approved drugs for patients in second line, so had only recurred from their disease for the first time. And they achieved these approvals demonstrating a 20% disease control rate in the first case, or for temozolomide, a 37% disease control rate. So what we're seeing here is actually higher than what the NCCN approved and recommended therapies are for patients with less previously treated disease. And I think it's really compelling that what we've shown here. Next slide. So how do we put survival in context for this particular trial? We've already said that the disease control is something that we're paying very close attention to and we're actually quite happy about. But as I mentioned previously, survival is really what is the game changer for patients with advanced brain tumors. And what we've shown here with the first 4 lines is there are 3 approved second-line therapies and 1 approved third-line therapy, demonstrating overall survival in the range of 3.7 to 7.8 months. Investigational therapies that are ongoing are showing even shorter, at generally 6.5 months in second line. And so what we're seeing here is roughly 7 months survival in anything that's been evaluated from first recurrence with approved and investigational therapies. And so anything that could show a survival that goes beyond 7 months would be something that we would be really excited about. On the next slide, here's where we have an opportunity to actually describe what survival looks like in a plot that we call a swimmer's plot. So just to orient you to this for a couple minutes, each lane of this plot, those letters A, B, C through K, represent an individual patient. The patients in green are those patients who achieved disease control, and the patients in blue did not achieve disease control. I want to spend just a minute or two on actually the patients in blue, because I think they tell us something really important. You can see that those patients, when they were treated, actually progressed and died within a very short time. And what that, I think, demonstrates to us all is that this is a horrible disease where progression and death is what would be expected in the absence of an effective treatment. And these patients likely had disease that was too far gone to ever derive benefit from what we could provide them. Alternatively, in the green bars, those are the patients that did achieve disease control. And you can see that although 4 of them have already died, there are 2 that are ongoing. So we will continue to follow those patients closely. Of the 2 that, of those 6 patients treated there, 2 of them actually maintained disease control for over 14 months and 1 of whom is still alive. So I think it's important to note also that the median overall survival was 9.9 months in those patients who achieved disease control, and that compares very favorably to the historical controls that we described earlier. So on the next slide, I just want to spend a couple minutes drilling a little bit deeper into those 2 patients with the prolonged survival of greater than 14 months. These patients would be considered by any standards exceptional responders. And the first patient was treated at a dose of 44 million cells. It was a third-line patient, so had already received 2 lines of treatment for their glioblastoma. And that patient achieved a survival of 19 months after 10 infusions, and actually went almost 80 days until there was relapse of the disease that was observed. But again, survival went out to 19 months. The other patient is a patient who received 88 million cells. This was a patient who had 3 prior lines of therapy, and that patient is ongoing in follow-up with survival that's beyond 14.4 months. This patient also received 10 infusions and was 70 days until that patient relapsed. So really exciting results from those 2 patients, and we believe that sets an important precedent for us to continue to deliver on. So a few words about safety, because this is, again, a first-in-human study where safety is primary. We were able to demonstrate across all the dose levels that were treated, there were no dose-limiting toxicities or adverse events that were attributable to the treatment that would actually cause us to not be able to dose escalate. So dose escalation proceeded without difficulty. An expected side effect that we sometimes see is called cytokine release syndrome in the context of cellular therapies. We didn't see cytokine release syndrome in this study. And tumor lysis syndrome, which is a phenomenon that we sometimes see in oncology with a rapid dissolution of the cancer, and that can cause important side effects that we have to deal with clinically. We didn't see anything of that sort. We did see a few grade 3 serious non-hematologic events, but importantly, none of them were considered treatment related. The cerebral edema that was observed in all 3 cases was observed in the context of disease progression, as were many of the other side effects listed there. So on the next slide, I can describe where we are, where we've been, and where we're going. So I think most of the audience will recall that in 2021, the phase 1 study was launched at City of Hope. And that really set this journey on its way. In June of this past year, we launched our own follow-on study to build from the learnings of that study. And in August, we press released the fact that the phase 1 dose escalation was complete. And today we're now reporting on the preliminary data from that phase 1 dose escalation. And I'm also really excited to let you know that we're going to be taking this study forward into a phase 1b dose expansion in a clinical trial in recurrent glioblastoma. So to summarize, we're really excited to be sharing this data with you all today. We believe this is very positive phase 1 data for patients with late-stage glioblastoma. The patients were generally 4th line and beyond, and were very difficult to treat patients, demonstrating a 55% disease control rate and almost 10 months survival for patients that achieved disease control. Importantly, there were the 2 patients I described with survival that went beyond 14 months, and the side effect profile was manageable at all the dose levels that were tested. So as I mentioned, we're excited to be advancing this program into our phase 1b dose expansion trial, and it's currently recruiting patients at Sarah Cannon Research Institute in Austin, Texas. And we're working hard to deliver on this program and really bring the value of the CHM 1101 to patients as fast and in as impactful of a way as we can. So we're really excited about where we are. We think this is an important asset, and we're looking forward to moving it further into the clinic. So on the last slide, I'll just kind of spend a few minutes kind of giving a summary of where we are as a company. So Chimeric is a small company, but we actually have 2 clinical trials now that are demonstrating positive phase 1 results. Our portfolio is innovative and diversified, focusing on areas of high unmet needs. We have 3 cell therapy platforms and 4 ongoing clinical trials. We're working with some of the best scientists and clinicians in the world. We're ready and building even increased readiness around technical operations. And we're really looking to maximize the value that we can realize with these different assets. And we're doing all of that really with a small team that has a great deal of experience developing cell therapies for cancer patients. And so with that, I think we'll start to take some questions.
Thanks so much, Jason. So hopefully, just before we turn to questions, Jason has been able to give you some more information about the exciting data that we announced this morning and really get across to you, how excited we are by this data, as it really exceeded our expectations in this phase 1 clinical trial. Now, with that, I've noticed that there are some great questions coming in. And so, Jason, I will start to knock off a few of those with you right away. The first is that it looks like from those 2 patients that had survival beyond 14 months, that being treated in 3rd line versus 4th line actually could result in longer survival. So does that mean using therapy earlier offers better results? And what determines the line of therapy that somebody actually is treated in?
It's a great question, Jen. Thanks for that. So to start, the observation is actually a really important one. With every line of subsequent therapy, we know that patients can be expected to derive less and less benefit. The cancers actually become just more difficult to treat. And so the goal at any time when you're developing a new medicine for cancer patients is to treat the patients in the earliest lines of disease that you can, because that's your greatest opportunity to demonstrate benefit. In this phase 1 study at City of Hope, the patients had oftentimes received several lines of therapy. And so our expectations would have been less for them to derive meaningful benefit. And so that's part of the reason we're actually really excited about what we're seeing.
Thanks, Jason. And perhaps this question goes a little bit along with that is, could this therapy eventually be used as an alternative to the primary or frontline treatment regimen?
That would be the hope, is that we can demonstrate activity to the level that it becomes a part of frontline therapy. But the way drugs get developed, you have to demonstrate safety and benefit in later lines and then move up with time once you can do that.
Perfect. Thank you. With this new data, could CHM 1101 see fast track designation from the FDA?
So we're actually really excited about potential regulatory interactions around this data set and what we're developing. And we'll be excited to announce any of those results as they happen. But yeah, we'll be in touch with the agency around how we can advance the program even more aggressively, and fast track is something that we do talk about.
Thanks, Jason. Does today's preliminary data release include data from cohort 3 and 4?
It does. That data is the less mature data set, though. So we have less information, but it is included.
And when you say less mature, Jason, maybe if you can just expand on that a little and what that means, particularly in survival.
Yeah, so we could only measure patients' outcomes based on the duration of time they've been under observation. So the more recently enrolled patients may not have achieved a milestone either in progression or survival that would be reportable.
Thank you. Is the phase 1b trial that is now open at Sarah Cannon Research Institute one trial or 2 trials when you talk about it being dose confirmation and dose expansion?
Yeah, so it's actually one trial. We're doing a very small cohort to demonstrate that the experience that they've had at City of Hope is also repeatable at the Sarah Cannon Research Institute, and so we'll be going seamlessly from that kind of initial cohort into our 1B.
Thank you. Now we're continuing the rapid fire for Jason here. Can you give us a sense as a clinician, Jason, with the side effect profile that we're seeing, what do you think your comfort level is when you compare it to things like radiation therapy or, frontline therapies that are being used in this area?
It's a really important question, Jen. So, like I mentioned, we always have to be thinking about safety first, and we want to make sure that the therapies that we're developing are first and foremost safe. In working with the team at City of Hope, it's become very clear that the adverse events that they're seeing associated with CHM 1101 are actually, for the most part, disease related and really not consistent with anything that would be concerning for this particular therapy. That being said, these are very sick patients, and so radiation, even oral medications, can be very poorly tolerated in patients that have not only a brain tumor, but all of the neurologic difficulties that go along with it.
Thank you. And maybe this question actually talks a little bit more about the safety profile. With that safety profile you're seeing, do you think there could be application for CHM 1101 in pediatric high-grade gliomas?
I do actually. City of Hope has treated patients that are younger than 18 on individual INDs. And I think the goal would be that we would be able to advance similar opportunities for children.
That's great. And so we have covered off the questions here, Jason. Maybe just one question, for you personally, seeing these results in glioblastoma patients, how do you see these?
I'm really excited, Jen. I have to tell you, and I think most people have been touched by cancer, and many even by patients with glioblastoma. And this is really one of the most feared diagnoses in medicine. And so we're now showing data where we're potentially modifying this disease and enabling these patients to live longer, better lives, which is something that I never could have imagined in patients with third, fourth, or fifth line GBM.
Thank you for sharing that. So, on behalf of Jason, myself, Matthew, and Paul, we very much thank you for taking the time to join us this morning to hear a little bit more about the data. We certainly invite you to continue to ask any questions if you have more questions about the data. We are incredibly encouraged by this. We very much are looking forward to advancing now the development of this asset towards those important registration pathways that allow us to actually get this medication out to patients, but also to be able to have better discussions about value realization of the assets. Certainly having clinical data that is now phase 1A clinical data and positive allows us to have, better and more meaningful discussions along those lines as well. So thank you very much. We are thrilled to be able to share this with you today and look forward to opportunities to speak again soon. Hope everybody has a great day.
Thank you all.